Tēnā koutou. Welcome everybody to the New Zealand Coastal Society webinar series. Now, we're pleased to be pushing this along again in 2021 after the fantastic success we had last year. Now we're lucky to have two speakers today, both PhD students who have some exciting new research to share with you all today. I will keep a record of questions and we can come to those at the end after the two presentations. So I encourage you to type in some questions in the chat box and we'll come back to those at the end. So our first speaker today is Zhang Xiao, PhD student from the University of Waikato, and he will be sharing some research today on the state of New Zealand estuaries using satellite-derived optical technology.
Hi guys, I'm Zhang Xiao from the University of Waikato. Today I'm going to talk about using satellite-derived optical properties to extract the true color in Tauranga Harbour. So water color is one of the most common water health indicators because it can reflect the overall water health. Except the sky-diffused radiations, the particles dissolved in the water play the most important role in color changing. So in general, water color is predominantly a reflection of its optical properties, in addition to the effect of dissolved matter and other suspended materials. So according to our previous research, we know that water color is correlated to CDOM, chlorophyll A, and other colored metal ions. So in order to measure the water color, we normally use the Fourier scale, which can reflect water color with different bands. But actually, it's only valid for the small scale. So in the large scale, we have to introduce the damped wavelength, which can be derived from satellites. The damped wavelength can reflect the optical color under the chromatic light very precisely. So in order to calculate the damped wavelength, we also have to introduce the chromaticity coordinates. The chromaticity coordinate pairs can determine the location of the point of interest in this horseshoe chart. So the definition of the damped wavelength here is the intersection of the line joining from a white point to the point of interest at the border of this horseshoe chart. So for example, the damped wavelength of this point is about 561 nanometers. So in order to calculate the chromaticity coordinate pairs, we also have to introduce the Tristan values, capital X, capital Y, and capital Z. The Tristan values can be converted from the remote sensing reflectance according to Warren 2018 with these three different calculus equations. So here, the Tristan values here function as the platforms to convert the reflectance to the water color. In our study, we would like to use the Sentinel-2 because Sentinel-2 can provide a highest temporal resolution since it revisits New Zealand every 5 days. It can also provide a high spectral resolution in 10 meters, 20 meters, and 60 meters with different bands. Totally, we can get 30 different bands, and we can use this band to calculate all kinds of normalized difference satellite data like NDCI or NDVI. And we can also use this data to estimate the concentrations of chlorophyll A and other suspended solids. So before we go to our study site, we have to figure out what happens after sunlight reaches the surface of water. According to Carnot's model, we have 4 different situations. Some sunlight can penetrate the water surface and get reflected by the water bottom and finally leave the water surface. Some sunlight can penetrate the water surface and get scattered or most scattered and finally leave the water surface. And some sunlight can penetrate the water surface and get reflected by the water bottom and have several internal reflections. Finally, they leave the water surface. But in our study, we mainly focused on the first situation because we know in the first situation is the most common phenomenon in the Gulf of the coastal waters. So here, we can see the water bottom type is the dominant factor to affect the reflection because the water bottom type decides the albedo. The bottom type mainly depends on the grain size. So our proposed study site should provide enough particle size information. So we are going to select Tauranga Hollow as our study site. We have 3 previous researches on sediments, which provide the full statistics of the particle size. Green, red, and blue dots here represent those 3 different datasets individually. So as we mentioned in last slide, the scanned reflectance cannot be directly used because the water bottom contributes too much on the water surface reflectance. Part of Tauranga Harbour is covered by the seagrass. The spectral signatures of seagrass also change the water surface reflectance and can change the water color as well. And part of Tauranga Harbour is intertidal, especially where we can see the water bottom, like this part. The water bottom of the seabed can also contribute a lot to the water surface. But actually, it's quite different for the deep channels because we know that most light can be absorbed by the volume instead of getting reflected in the deep channels. So we do not have to correct the reflection for the deep channels here. And then we can have a look at our type law. First, we are going to use normalized difference water index to extract the exposed regions and inundated regions. For the exposed regions, which is really good because they're always exposed, they are sediments, and we do not have to correct them. And for the perpetually deep water, it's also good. According to our assumption, the water bottom and the diffuse attenuation coefficient are assumed not to have a significant effect on water surface reflectance. So we do not have to remove them as well. So in our study, what we have to focus is intermittently inundated regions and perpetually shallow waters. For the intermittently inundated regions, sometimes they get exposed, sometimes they're not because of the different heights. So when they are at low tide, we can directly get the remote sensing reflectance of the water bottom. But for the perpetually shallow waters, they're always underwater. We cannot get it directly. So we have to estimate the water bottom reflectance at these conditions. Here, we build a regression between the particle size and reflectance to finish the estimation. But it's impossible for us to get the particle size in each pixels. So what we have to do is build another 2D regression between the particle size, water depth, and this distance from the entrance to the pixels we selected. And finally, we can correct RW. So in our procession, we would like to remove the water bottom. We are going to use the Lazingas model, where R is observed reflectance from the satellites. RW is the true water color, true water reflectance. RB is the water bottom reflectance, and K is the diffuse attenuation coefficient, and Z is the water depth. Here, figure A shows the shape or the nature of this curve. What we can see here is once the seabed is exposed, the RB does not change anymore, and in the inundated regions, the reflectance goes down with the increase of the water depth or the elevations. Here we just assume the RB is about 0.11, RW is about 0.023, and K is about 0.4. And we also select a transect across the Tauranga Harbour as an example. So figure B are the examples of the observed relationship between the reflectance and the elevations. The elevation of pixel is relative to mean sea level from a case study site in Tauranga Harbour. So in reality, we can see in different tide status, the reflection may be different. The reflectance at the low tides is significantly higher than those at high tides. So in order to calculate RW here, we have to focus on proximal pixels here. And we have to set another assumption that K is not changing with water depth. And then we can rewrite our previous equation to this new one. According to this new equation, we can start to guess the true reflectance, RW, And each RW will have a corresponding K. According to our assumption, the slope between K and Z should be 0 because we know that K does not change with the water depth. We can also plot the variances of K and RW at this time, and the minimum values of variances has the greatest likelihood of approximating the true value of RW. Here we just take an example in band 2. So the correct RW in band 2 is about 0.023. If we would like to use our models, there are several requirements. First, the underlying water properties and seabed properties should be very uniform over the region of proximal pixels. That is to say, we have to conduct a manual check on the selected pixels. We have to avoid the pixels with seagrass and other vegetations. Proximal pixels can either be selected from a single image, where the water depth varies spatially, or can be selected from a series of images collected at different times. So in the first case, we can get a set of the images with high temporal resolution but low spatial resolution, vice versa in the second case. And then we can have a look at our results. First, we build a 2D regression to estimate the particle size in each pixel. So what we can see here is the water depth has the most important role in determining the particle size. And if we visualize this data in northern and southern Taronga Harbour, we can see only in the deep channels, the particle size has a significantly higher values. But for the intertidal regions and shallow water regions, the sediment particle size is quite similar to each other. So if we would like to simplify our steps to get RB, we can probably treat the sediments of the intertidal regions and the shallow water regions very homogeneously. And once we get the particle size for each pixel, we can build another regression between the particle size and RB. If we check the scatters on the right figures, we can see in band 2, band 3, and band 4, they probably can show the same fitting model. So here we build a simplified log regression with different A, B, C. According to the R-squares here, we can see the fitting model, the fitting quality is not really bad. Even we know that optical properties of sediments does not really change very greatly in 3 years, but we do divide them into 2018, 2019, and 2020 3 groups to get a better accuracy or understanding of our model here. Once we calculate RW, we can convert RW to our dumped wavelengths. So in the figure A, we can see the dumped wavelengths of the color of the exposed sediments, which we do not need to correct them. And in the figure B is the water color of the deep water channels and the shallow waters before we remove the water button reflectance. And figure C shows the dumped wavelengths of the shallow waters after we remove the water button reflectance. So if we compare figure B and figure C, we can see the dumped wavelengths after we remove the water button reflectance is significantly lower than before we remove the water button reflectance. And the color of the water in the shallow water is greener, and the decrease of dumped wavelengths is about 20 nanometers. Figure D shows the water color of the deep water because we do not have to remove the water button reflectance for them. So figure B and figure D has the same values for the dumped wavelengths. And if we scatter this data in the previous horseshoe chart, we can see some scatters at the edge, green like this part, have been corrected because we manually select the pixels without seagrass or other vegetations. The dumped wavelengths of pixels from shallow waters regions like this part, they're clustering together because we are using our model to decrease the spectral resolutions and remove the water button reflectance. And the dumped wavelengths of the sediments, they do not change a lot because we don't have to correct the reflection for them, and the color should be all the same before and after remove the button reflectance. And then we are going to talk about the scatters into different months. Here, we just take Southern Taronga Harbour as an example, because I compare the Northern and Southern Taronga Harbour. The trend are quite similar. The only difference is the water color in Southern Taronga Harbour is green, a little bit greener than North. So if we have a look at the exposed sediments in the Southern Harbour, we can see they're always not changing very greatly. That means there's not changing pattern for the exposed sediments. But if we check the dominant wavelengths for the deep waters and shallow water, especially for deep water, we can see there is a obvious seasonal fluctuation. The dominant wavelengths of the deep water decrease to the minimum values in winter and returns to the peak in summer, which means in winter the color will be greener, will be bluer, and in summer the color will be greener. And if we also check the standard deviations of the shallow water, what we can see is it has the greatest standard deviation because we are using our model, and it can probably cause some errors here. And then if we compare our water data with other water health indicators, we can find the chlorophyll A plays the most important role in color changing, because we know that with higher concentration of the chlorophyll A, we usually have a greater possibility of the greener water. Clarity is also important to change water color because it can change the water environment, especially in the penetration of the light. Conductivity, pH, salinity can also change the water environment, but not in a very direct way to change the water color here. And then we can plot the diffuse attenuation coefficient in band 2, band 3, and band 4. The diffuse attenuation coefficient is the factor to evaluate the water color turbidity. The tidal conditions seem to not to have a strong effect on KD in 3 bands, and the seasonal fluctuation of KD is not really apparent because the increasing and the decreasing trend may occur in all kinds of seasons. If we compare the K values of Northern and Southern Taronga Harbour, the values are quite similar. Probably the Southern Taronga Harbour has higher values. But if we compare the K-PAR data from Mariana's work with ours, it shows our result is quite reasonable because K-PAR values run between K in blue and K in red. And the future work will be extend the research to Ohiwa, Raglan, Kaipara, and other estuaries. And we will also have to clarify the correlations between the water color or KD and other ecological biological factors. We also need to classify the New Zealand estuaries with water color. And finally, we are going to explore the possibilities of evaluating the estuary state with water color. That's all for today, and thank you so much. If there is any question, will be super.
Thank you very much, Sheng Xiao. I cycle my bike into work over the Tauranga Harbour every day, so it's great to see the Tauranga Moana being used for this really important research. I think if you stick around, Sheng Xiao, we'll have some questions at the end. Thank you so much for that presentation and for sticking to time. You're even a bit early, so well done. Okay, I'd like to introduce our next speaker now. His name is Inigo, and he has come to New Zealand from Spain to study fine sediment effects on seagrass. Outside of his study, he enjoys scuba diving and marine conservation. And I think he's actually also worked as a fisheries observer. So I'm sure he's got lots of stories to tell. But yeah, I'd like to hand over to you now. Thanks, Inigo.
Hi everyone, kia ora. Welcome to this second talk of the day. And today I'm going to be talking about fine sediment effects on seagrass Tostera muyeri, which is the topic of my PhD and the project that have kept me busy for the last 3 years and a half. So yeah, as probably our backgrounds are slightly different, I'm going to give you a few statements about seagrasses in general. So letting you know that seagrasses are a unique group of flowering plants that have adapted to exist submerged in the sea. They are distributed across the globe, but unlike other taxonomic groups with worldwide distribution, they exhibit very low taxonomic diversity. There are just about 60 species around the world compared to the 250,000 terrestrial angiosperms. And they come from 4 independent lineages that evolved from a single monocotyledonous flowering plant about 70 to 100 million years ago. And they have successfully colonized all but most polar seas, and we have the southest point of growing seagrass in the planet here in New Zealand, in Stewart Island. So since we have been probably all of us without being able to travel around much, I'm going to bring you into my personal seagrass journey. So as Mark said before, I come from Spain, and my first connection to seagrass was in the West Atlantic Ocean with the Thostera marina plant that looks like what you can see there in the image. And then I moved into the Mediterranean Sea to work on some Posidonia restoration projects. Posidonia is the biggest plant that you can find in the Mediterranean, and it looks like that. It creates a massive seagrass meadows. And we also work on another species such as Cimodothea nodosa, which is considered the smaller cousin of Posidonia. And in my way to New Zealand, I was lucky enough to stop in some of the tropical locations with seagrasses growing, and there are very different species. The one that you can see in the top is Alophila hovalis, which is this round kind of leaf species, which is really beautiful to see. And what you can see in the top in the bottom is Enhalus acroides. And in 2017, I arrived in New Zealand, and this image will be more familiar to all of us, and it's an intertidal seagrass in Wharekawa estuary. And if we look further in detail into our plant, we can see the next pictures. So that's how seagrass looks like, and maybe for some of us is we are more familiar with what's happened below ground, but maybe not for all of us. So if you take the plant out of the substrate, you can see that it's like an iceberg. There are a lot of things going on, and they have a very well developed below ground structure of roots and rhizomes. So what you can see in these two images, in the top is Whangarei Harbour intertidal meadow, and in the bottom is Pawatahanui Inlet under submerged condition. And these seagrasses are key, very important habitats. So I'm going to start by telling the main key ecosystem services that seagrasses provide to our coastals. So the first one is that they are able to give coastal stability and reduce the hydrodynamism from the waves and unidirectional flow, and they may be used as a buffer for the sea level rise in the future. They do also provide blue carbon ecology, which is a hot topic right now. And basically what this means is that seagrasses are able to use the CO2, which is in the atmosphere, to perform photosynthesis, sequestrating carbon in the soil and helping us to deal against this stressor for the society. And they are also a very important primary productivity for coastal ecosystems. They are not as famous as rainforests and other, very important ecosystems, but they are, along with the mangroves, a very, very, very key primary productivity for our coastal ecosystems. And in terms of fisheries and nursery habitat, they do also provide a very important habitat for species like snapper, stingrays, etc., and also for a lot of invertebrate species being the base of the ecosystem. And they do also help on the maintenance of water quality. And I think Sancho has been covering a lot of this stuff before pretty well. And the seagrass is able to trap sediments and provide a higher water clarity. And as we were talking before, there is a lot of stuff going on below ground, and seagrass rhizosphere and rhizome areas are a very important area where some key ecological processes occur. And overall, the take-home message is that if the seagrass is thriving, our invertebrates will be happy living, as you can see in the top of the of the slide, and our economies will be healthy, and our lands, of course, will be healthy too. But unfortunately, that's not the case, and seagrass is declining all around the world, and that's a huge loss. So what you can see in this plot here shows the in red the areas where seagrass has been reported to be declining. And as you can see there, in New Zealand is not an exception, and we can pass from a very beautiful intertidal meadows to bare mudflats, losing all of these ecosystem services that seagrasses provide. And that's a huge thing, and it should be enough to worry us about the importance of the problem. But we as humans like to always put things in balance. So I'm going to give you just two average numbers, which should be important. So seagrasses are estimated to provide about 1.9 trillion US dollar per year, which is a massive amount of money. They are being lost at the rate of 110 kilometers square per year. So you made the maths. So when the pocket is touched, I like to see that probably not just the scientists, but the whole communities will be really worried about the problem that we are talking about today. So in terms of New Zealand, what you can see in this map is the areas where seagrass is being reported. And the Department of Conservation has been working on creating these seagrass and mangrove extent maps that are available in their website. It's very useful. And I would like to introduce you this mobile phone app, which is called Seagrass Spotter, that will be able for us to provide to create better maps. You can just with your phone take note of where you have seen seagrasses and put them in the system so we can have access to it, so we can improve our data. And what the situation of New Zealand seagrass is the next. As you can see there are two harbors where recent increase has been reported, which are Manukau and Kaipara Harbor, whereas there are quite a lot of harbors with a significant decline that over the years that's been reported, such as Tauranga Harbor, Whangarei, Bay of Islands, and also Porirua Harbor. And there is also a big gap on the information we have from the past from the seagrass meadows that were growing before, and they are not growing anymore. So it is a gap there, but it's definitely we should take care of our seagrass because it's a current trend globally and also in New Zealand that it is declining. So there are a lot of seagrass loss mechanisms, and there are natural seagrass loss mechanisms such as extreme climatic events or wasting disease, etc. But the main problem is us, it's anthropogenic effects. So there are introduced species, global climate change, physical damage, eutrophication, and today I will be talking about fine sediment pollution. So for us, it's not very rare to see these kind of images where we can see the problem that's going on upstream and how we receive it in the receiving environments in the estuaries or in the coastal areas. So in order to explain how fine sediment affects seagrass, I've grouped them in three groups. The first one is the time where fine sediment is suspended in the water column, and hence it still slides from the water columns for the seagrass. So that makes light not available for performing photosynthesis. Then the second group of effects is when this fine sediment is settled on the leaves of the seagrass or in the substrate, and it keeps on to smother seagrass, provoking anoxia and also impeding the correct amount of light to reach to the seabed. And the last group of effects is when these fine sediments are intruded into the substrate and provoke further problems in the rhizosphere, such as anoxic condition and high sulfite concentration phytotoxins. And the fact that I have put them in three groups, it doesn't necessarily mean that they are not occurring at the same time or interacting between them. So in order to further study the fine sediment effects on seagrasses, we ran a case study in the Pawatahanui Inlet. It is a harbor which is 20 kilometers north of Wellington Harbor. And what you can see there in red points is the historical seagrass habitat, which is areas where seagrass used to grow before. Then what you can see there in green is the existing seagrass habitat. And what you can see in blue is the potential seagrass habitat, which is areas where seagrass used to grow before and is close to existing seagrass, and it's been coming and going from this area. So it's an area which is potential for seagrass growth. And in this harbor, we recorded a loss of 39 hectares of seagrass since 1980s. So we measured the available light and substrate properties within these three habitats and make a comparison between them. And what you can see in this plot is the main result of it. In the x-axis of this PCA, you can see that the historical seagrass habitat tends to have a much higher substrate muddiness and a much higher total suspended solids, whereas the existing seagrass habitats have a much higher PAR, or photosynthetically available radiation and a less anoxic substrate. And what you can see that in the y-axis is the differences within the sites, which are mainly driven by the poor water chemistry in terms of phosphate, ammonium, and hydrogen sulfide. And the fact that the clustering of the potential seagrass habitat and the existing seagrass habitat is slightly separated, maybe due to the fact that seagrasses are able to modify their environment with positive feedbacks, such the ones that we have been talking about with their ecosystem services. So the main highlights of this case study are that fine sediment is behind estuarine intertidal seagrass decline in Porirua Harbor, that the seagrass mud follow a saturation relationship with an optimum of 13% mud, that we didn't find a substrate with a higher percent of 23% mud with seagrass growing on it, and the damage mechanism, our hypothesis is that the anoxia is the main damage mechanism, and the intertidal light levels were always above the determined threshold for seagrass to grow, but it wasn't still growing in the historical habitat, which made us, we needed to rethink on the consideration as light as the main issue and think on other substrate properties, and we did publish this article on the Journal of Marine Science and Engineering for further information if you are interested on it. And after working on this, we have the question if does the interaction between the substrate condition and irradiance, when this is close to persistent threshold, control the seagrass growth and persistence. So in order to try to explain this question, we made a factorial aquarium experiment with two treatments of light, with a low light level and very low light level, being both above the thresholds for seagrass survival. And we used two different substrates under those light treatments, the substrate which was existing seagrass substrate, a healthy substrate, and historical seagrass substrate, a substrate which has been modified through time and is being deteriorated. And the main hypothesis was that seagrass wasn't able to actively pump oxygen through the historical seagrass substrate in order to deal against phytotoxins such as sulfide and stuff. So we exposed seagrass for 6 weeks on this experiment, and this is, of course, easier said than done. We needed to work a lot and do the experiment twice in order to get to the results. And once we got to the main results, are these ones as follow here. What you can see in the top left plot is the increase in shoot number of our planted seagrass, and what you can see there is that the increase in shoot number is mainly driven by the effect of light, and also the effect of the substrate, and the interaction between both. And what you can see in the top right panel is the rhizome growth. This is the below ground structures growth. And as you can see there, the only treatment which show a negative growth was the one with existing with historical seagrass substrate and a very low light level, which is really key result for our study. And we did measure with this micromanipulator the oxygen profiles of the substrate through time, with the idea of we were going to pick this as a main mechanism for seagrass not to respond to the substrate, but we actually couldn't. And what you can see there in the y-axis is the sediment depth through week 1, 3, and 6. And as you can see, the difference between the profiles are run by the light treatment, not by the substrate treatment. And during the course of the experiment, this difference attenuates to the fact that there is actually no difference between them. So we couldn't say that the oxygen is the main mechanism running the explanation for the treatments. And the main highlights for this study are that modification-related multiple stressors and irradiance, when this is close to persistent threshold, interactions control seagrass growth and persistence. The seagrass light requirements are substrate condition dependent, which is a very key result. And photosynthetically available radiation thresholds for seagrass persistent may not fit for purpose if interactions with substrate condition are not considered. This is like the iceberg. If things are going down below ground, we need to consider them also. they are pretty important because they are interconnected. So we may be overestimating PAR thresholds, and we must take into account the legacy effects of the fine sediment already incorporated in the substrate. And multiple stressors are interacting to affect our seagrass growth. And moving on from sediment a bit, we were working on a restoration experiment, and we were unlucky that we did found Chaetomorpha ligustica, which is filamentous algae which grows over the seagrass. And what you can see there is another threat for seagrasses, which are already under a lot of pressure. And this growth of this algae was smothering seagrass in one of our transects. And we were wondering what kind of a species it was, because it's quite hard to identify. So we take some samples, bring them to Roberta Darkina, which is a NIWA Wellington taxonomist, which helped us in this project to identify this. And we realized that it was Chaetomorpha ligustica, and we reported for the first time the effect of this algae on seagrasses. So what you can see there is the effect of it before seagrass being exposed to it and after, and it's quite impressive and not good for seagrass. We were able to quantify this in one of our transects, and what you can see in the plot there is the transect that was really affected by Chaetomorpha showed a decrease of 50% in the cover of seagrass, whereas the one that was unaffected didn't show any difference. And we attribute this to the fact that this Chaetomorpha growth was smothering seagrasses, as you can see in that image there, and created a very anoxic layer, plus deprivating seagrasses from light. So coming back to the sediment, I would like to show you this as a multi-stressor model, and fine sediment acts as a very complex pollutant to affect seagrasses, both physically and chemically, as depriving seagrasses from light and creating a very challenging rhizosphere in terms of a very high bulk density and very low porosity of the substrate, which doesn't allow seagrasses to actively pump oxygen to protect against sulfite and other phytotoxins. And all of this interaction net drives the state of a seagrass ecosystem. So what you can see there in the plot is in the y-axis, you can see the seagrass cover, and in the x-axis, you can see the photosynthetically available radiation. And seagrass ecosystem may move from state 1 to state 2. So state 1 is the vegetated state where everything is healthy and everything is looking good, and state 2 is the bare and unvegetated ecosystem where seagrass is not present anymore. So in order to further understand what are the trigger points or the important factors that run these ecosystems to move from one state to the other, we should definitely consider interaction in terms of fine sediment, but also in terms of other stressors that will complicate the thing quite a lot, such as eutrophication or other microalgae growth, etc. So there is, and it should be, some light. You may have perceived that my vision is a bit pessimistic, but there is, and it should be, some light. The seagrass restoration is possible, and conservation is possible too. We just need to put our best effort on improving our agricultural and urban land water management, making some riparian planting to buffer what's going on into the streams to end up in the estuaries. And we need to definitely improve transplantation guidelines with 8 go, no go, or site selection procedures, with the new knowledge that we are incorporating in terms of fine sediment, nutrients, light, etc. And of course, in order to restore the habitat, we need innovative interventions to tackle the substrate legacy effects, which are already in place in some of our estuaries. And I would like to acknowledge NIWA and University of Waikato for giving me the opportunity to work on this project, which has been really, really cool. And also my supervisory team, Flair, Ian, and Marilyn, thank you for your support, and a lot of people that's been helping me out through these 3 years. And I'm happy to take questions as long with thank you.
Thank you very much, Inigo. That was outstanding presentation. this stage, I don't see any questions. So I think you've both nailed it. So, it's a good warm-up for defending your PhDs. You've managed to keep the crowd pretty quiet. So I'll just wait a couple of minutes, to see if any questions come through. But I do recall one time walking along Tauranga Harbour with my son. The coastal walkways, you can see the seagrass. It's quite close to the coast. And I recall one time seeing a stingray just float by with a kingfish following behind it. So that ecology that it brings in close to the coastline is amazing. For such a small plant, yeah. I had a question, Inigo.
Yep.
You mentioned some areas where seagrass is declining.
Yep.
Is that being replaced by other species like mangroves, for example, or is it just becoming dead zone?
Yeah, it is in some of the estuaries. The problem is with fine sediment coming in and making seagrass unable to stay there or recolonize new areas. And there are some other problems with, rather than with mangroves, with ulva growth that is been impeding seagrass to stay too. But yeah, mainly I would say that the problem are maybe the eutrophication and fine sediment inputs in estuaries, but every estuary is different. So in the case of the Powhatan Inlet, which I'm more familiar, is definitely fine sediment going on in there. And also we have some big problems with ulva in Tauranga. Yeah.
Okay. Yang Xiao, in regard to your technology, could it be applied to large river type environments, or is it not suitable for flowing water?
Yeah, actually, this topic is from like the rivers, the catchments, and it's extend to the coast waters because the coast water, we know that some coast water has very apparent one, but sometimes they are very turbid. So what we would like to do is do this first and come back to water and do like a large monitoring, including water, rivers, and coasters. So it'll be like a large monitor work.
Okay. Thanks for that. I do have one question here for Inigo. Can you tell us about the restoration efforts overseas?
Yeah, sure. So the part that I'm more familiar with is the restoration effort that's going on in the Mediterranean Sea. So there are some huge projects going on in the Balearic Islands in Mallorca, and it's a lot of electrical companies that are funding this in order to improve. And from restoration perspective, we can improve in both ways. We can improve the asexual reproduction and sexual reproduction of the plants, which hasn't been done in New Zealand yet. And we can use clonal growth in order to do that, or we could use the sexual reproduction through seeds. And we haven't done that in New Zealand yet. And it's a technique that's been pretty much used in Australia too. So we're looking forward to see if we can get to improve our restoration in New Zealand. We've done some trials in Whangarei Harbour that did work, and we did manage to restore some seagrass habitat, and we should be able to try to apply this to different estuaries such as Tauranga, Porirua, etc. It will be really cool.
Excellent. I think you can see that follow-up question there. Can seagrass restoration be used in carbon offsets? Do you know anything about that?
Yeah, it's definitely a very important ecosystem services that seagrass provide, and it's definitely worth it to actually restore seagrass in order to get that carbon offsets to be replaced. And it is a very, very important thing that we could do. Yeah.
Fantastic. Okay. Now, Yang Xiao, I have a follow-up question there from Colin regarding your method, which you showed the site study being Tauranga Harbour. And I think it's just a follow-up question to confirm, does your method still work in disturbed free surface with flowing water within the coastal environment?
Yes, that's a good question because actually we just focus on a disturbed free surface, but we would like to, sorry. Yeah, it can work with the disturbed free surface, but actually it does not really work well with the flowing water because we know that we have to remove all the other stressors or other factors which influence the water a lot. So currently the model is only valid for disturbed free surface water, like the coastal regions or the catchments or the lakes and the open sea as well.
Thank you.
Thank you, Colin.
Okay, I think that's all the questions we have today. So I'd just like to thank both of our speakers, both from the University of Waikato. So I think you've set the bar very high and put the challenge out to the other students who are listening in today to share some of your results at future webinars. I would also like to encourage everyone to consider joining us at the Coast and Ports Conference later this year in Christchurch. It's in the end of November. And I guess another plug for the app too that Inigo mentioned. Just want to remind us what the name of that app was for spotting seagrass and mapping that.
It's Seagrass Spotter. Yep.
Seagrass Spotter. Fantastic. Excellent. Well, I'd like to close there. So thank you all for attending, and thanks again to our two speakers and to Engineering New Zealand for making this happen. Thanks, and have a great day.
Thank you.
Thank you very much.